video is going to explain what a Punnett square is and how to use it to get full marks in an exam question. A Punnett square is a type of genetic diagram. It is drawn out with 3 columns and 3 rows, as shown here. Columns and rows. Its purpose is to predict how a characteristic would be inherited by the offspring of a particular couple. I'm going to take you through using an example characteristic. Whether or not your earlobes are attached. There will be a gene on a particular chromosome in your cells that controls whether this happens or not. The location of this gene is called the locus. It will be on the same place on the mother's chromosomes and the father's. Remember, all chromosomes come in pairs. Let's say this is the location of the gene. The mother will have 2 copies and the father will also have 2 copies because their chromosomes are in pairs. There are 2 versions of this gene called alleles. There is one allele for having a detached earlobe and there is another allele for having an attached earlobe. Let's just give them colours and letters. When you're talking about a characteristic, you always have to give it a letter of the alphabet. This must be a letter that looks different in capital and lowercase, so C is not a good choice. The letter could relate to the characteristic, but it doesn't have to. In this case, we're going to use the letter E. So, a detached earlobe is going to be given a capital E, and an attached earlobe is going to be given a lowercase e. Capital letters are always given to the dominant allele. This is the allele for which you only need to have one of in order for it to be expressed. A lowercase letter is always given to the recessive allele. This is the allele for which you must have 2 copies of in order for the characteristic to be expressed. In this example, let's say both parents have detached earlobes. This is what we call their phenotype. It is what the characteristic that you can see. But let's also say that they have 2 different alleles. So let's say the mother has a big E on this chromosome and a little e on this chromosome. Similarly, let's say the father also has a big E on this chromosome and a little e on this chromosome. Therefore, the genotype, which is the 2 letters that you have, are different. One capital and one lowercase. When we have 2 different letters, 2 different alleles for a particular gene, we call this heterozygous. So both parents in this example are heterozygous. Now, remember, in order to produce gametes, which are sex cells, both parents will undergo the process of meiosis. Mark it with a star here and a star here. In meiosis, pairs of chromosomes are separated, and therefore the 2 chromosomes will go into different gametes. So we also have to separate the alleles. Big E will go into half the eggs the mother makes, and little e will go into the other half. Similarly, for the father, when he undergoes meiosis to produce sperm cells, 1 chromosome from this pair will go into half of the sperm. They will get the big E, and the other one will go into the other half. They will get the lowercase e. Remember, gametes fuse randomly. So, the white squares in my Punnett square in this video show possible combinations in the zygote, or the fertilized egg, and therefore possible combinations that the baby or child will have for this particular characteristic. So, we must match up all possibilities, like so. We take this particular egg, fusing with this particular sperm, we're going to get the big E from the egg and the big E from the sperm. But what about if this particular egg fused with this particular sperm? We're going to get the big E from the egg and the little e from the sperm. What happens if this particular sperm fuses with this particular egg? we're going to get the little e from the sperm and the little e from the egg. And the last combination is if this type of egg fuses with this type of sperm, we're going to get the big E from the sperm and the little e from the egg. These are all the possible zygotes that this couple could produce upon fertilization. Finally, we have to do two things with the results. We have to identify the phenotypes, and then we have to express the probability or the chances of each one. A phenotype, remember, is the physical expression of the genotype. So the phenotype is the characteristic. So we have to identify which offspring are going to have detached earlobes and which are going to have attached earlobes. In the example shown below, the genotypes big E and big E little e will both produce the phenotype of detached ears. So we need to identify this clearly in our answer. In this example, the genotype little e will produce attached earlobes. It is the only genotype that can do this. Remember, with a recessive allele, you need two copies in order to have the characteristic expressed. So, we've done our first job. We have expressed the phenotypes. Now we need to actually state the probability of having each of them. So, if you have a look at it, I'm just going to go round all of the genotypes that could produce detached earlobes. Remember, big E or big E little e. And then all the genotypes which could produce attached earlobes, just little e. So, probabilities can be expressed out of 4, or they can be expressed as a percentage. So, let's fill in the table I've drawn. What is the probability that their offspring will have detached earlobes out of 4? Well, attached earlobes is only this one here, 1 out of 4. What is the probability that their offspring will have detached earlobes? Again, it is this one here, and this one here. That is 3 boxes out of the 4. So we say there is a 3 in 4 chance. Percentage-wise, 100% would be all 4 boxes, therefore 1 box represents 25%. So again, for attached earlobes, there is only 1 box, therefore there is a 25% chance. And for detached earlobes, there are 3 boxes, therefore we say there is a 75% chance. You do not have to use both methods, but you must choose one. Either express the probability out of 4 or express it as a percentage.